Good morning. Well, it's morning for me. You might be watching this in the evening, so good afternoon or good evening if that's the case. We're going to have a segment of our unit 3 in the beginning here that's nerve physiology. So if I take a peek at the syllabus, we're going to have 3 lectures on nerve physiology, and then we actually have spring break. And you've got no assignments due over spring break. Honestly, it's a great time to study a little bit of the nerve physiology that we're going over right now. And, just recognizing that there are mechanistic processes on action potential that students really tend to wrestle with. And so I want to make sure that you hear that ahead of time. You take a peek at chapter 7 and prepare for this coming week. That we've got the last week before spring break. And then over spring break, I know you probably have really exciting plans, as do I, but I'm hoping that you can carve out a little bit of time just so it doesn't go dormant. You don't forget about what we've gone over. And then when we get back from spring break, we've got central nervous system, which is the brain and spinal cord, and that's going to be our unit 3. So we'll have nerve physiology, and then we'll talk about the brain and the spinal cord. And then we'll have unit 3 completion, which is preparing you for exam 3. So that's the schedule. So what I'm going to do right now is I'm going to share my screen, and we're going to get started. So neurons, you can see in the upper left some images of neurons that are kind of making these synaptic connections. Pretty dramatic, pretty cool. And the nervous system is really a physiologic system in our body that really helps to control things at very rapid levels or rapid speeds. And then we have in the bottom right a neuron, and you can kind of appreciate the different parts of the neuron. You can see the cell body here. So this is essentially a cell. So if you think about to cellular biology, we have many components of the cell that are represented here. And we're going to go over that today. And there's some specialized structures, some unique structures like the myelin sheath, which are these sort of oval looking structures that extend down the axon. And we'll talk about its purpose. Okay, first up, let's take a peek at what neurons do. Well, the whole point of a neuron is really to send a signal. That's its goal. And there's a overarching architecture. And what you're seeing in this slide is you're seeing the peripheral nervous system shown green and the central nervous system shown on the right side in blue. And these neurons connect our outside environment to the central nervous system. And then the central nervous system processes that information and sends a command to the periphery to do something. So we've got step number 1, we've got a sensory, or what we refer to as an afferent with an A, neuron that collects information from the outside environment, sensory perception, sends a signal. You can see the arrow sending the signal to the central nervous system. In the central nervous system are what we call interneurons that are also referred to as association neurons. And what they do is they are confined to the CNS, the central nervous system, within the brain and the spinal cord. And they send the signal from one location to another, depending upon, is it the sense of smell? Is it input about vision? Is it heat, sense of touch, whether it's light touch or maybe trauma? So that interneuron pathway sends it to different parts of the brain. And after spring break, we're going to look at the central nervous system and the different places that signal will go. And then you do something with the response. You send that signal back out into the periphery via motor efferent with an E, output neurons, and it goes to effector organs like muscles or glands. They're going to go do something. A gland would be like a sweat gland or maybe a digestive gland that is going to release an enzyme to help you digest food. So this is really the blueprint of what we're going to be looking at. So now, how is the nervous system organized? So the nervous system is organized really into two main branches. We've got our central nervous system, which I've already really kind of mentioned. That's the brain and the spinal cord. Refer to that as your central processing unit, or like your CPU for those of you that that like computers and understand computer language. The central processing unit, this is the command center. This is what determines what's going to happen. And the brain and the spinal cord, so you can see in this picture on the left, the brain, as well as an extension of the brain known as the spinal cord, make up the central nervous system. And then the peripheral nervous system connects body parts or segments around the body that are going to be interacting with the outside environment, whether it's external stimuli or whether it's going to have you do something within the outside environment. And spinal nerves and cranial nerves and nerves in the periphery, like in your leg and your arm and your trunk, these are all part of the peripheral nervous system. And we're going to look at the peripheral nervous system in more detail today. And we're going to further subdivide it into different categories. So on the left side, you can see the central nervous system, very simplistic, brain and spinal cord for today's purposes. And then after spring break, we're going to unpack this. But today and into next week, we're actually going to be in the peripheral nervous system. And we've got sensory and we have motor. And we already kind of mentioned these terms. We've got sensory afferent with an A, and that takes information from sensory receptors. That's that first blank. And it sends it to the central nervous system, or the CNS. That's the next blank. We call these either somatic afferent fibers, and those are usually from like the skin or muscle or joints. And then visceral afferent fibers are from the viscera, which is a word that means like soft tissues, organs of the body. Those are in the abdominal cavity as well as the thoracic cavity. So all of that information is going to the central nervous system. So you're really looking at this left side here, visceral, somatic. Now we look at the right side and we have kind of the same thing. We've got motor efferent sending signals out. So it goes from the central nervous system to the effector organ, right? And that effector organ could be a gland or a muscle to tell it to do something. And now we can look at, further dividing this into the nervous system as far as the somatic, and looking at visceral, where we've got autonomic referred to as visceral. So it's another terminology for the visceral motor system, right? And we divide the visceral or the autonomic. That just is another word that means like automatic, like you involuntarily control it. You don't voluntarily control it. The somatic nervous system is under voluntary control, and the autonomic is under involuntary, subconscious. You don't have to think about it. And this happens into two divisions shown at the very bottom of the tree, one being sympathetic activity and the other being parasympathetic activity. And the way to think about these two or categorize these two is sympathetic are things that will fall under the category of flight or fright or fight. Okay, so the three F's. So either you run away, right? You're startled, you either run away or you stay and you fight. Any one of those activities, you're going to raise heart rate, you're going to increase respirations, you get amped up to go do something, whether it's fight or whether it's run away. Parasympathetic activity is categories in terminology known as like rest and digest, where you're going to kind of relax, slow things down, maybe think about taking a nap, or as you wind down the evening and you fall asleep, your parasympathetic activity is what's dominating, where everything is actually starting to slow down. Metabolism drops, respirations drop, heart rate drops. So this is really our overarching roadmap of where we're going to be. Now let's move into some of the physiology. And it isn't until the later part of the lecture that students, as I mentioned, kind of get into the challenging aspects of action potential. And we're going to introduce action potential today. And I'll warn you before we get there, everything up into that last point is usually pretty straightforward. And then in our next lecture, we're going to dive pretty heavily into action potential, and that's going to be a bulk of what it's going over. So you're going to see on your quiz for chapter 7, you do need to read the chapter, and you need to make sure that you're familiar enough with action potential and how it works to answer a few questions on the chapter quiz. Okay, so let's look at nerve physiology, and let's look at cell types. We've got 2 main cell types in the nervous system, whether it's in the central nervous system or the peripheral nervous system. The first cell type is what most people are more familiar with, and those are neurons. They're excitable cells. What does that word mean? Excitable. Well, they will initiate, or their resting membrane can be electrocuted. They can be excitable. They can be energized, and they're energized with electricity. And you're thinking, electricity? Well, we're talking about very small amounts of electricity. We're talking about individual ions like sodium and potassium and chloride. And we're going to unpack that. And because like sodium carries a single positive charge, if you had a bucket of sodium, so to speak, you had 20 sodium molecules, you might get 20 positive charges, but it's going to be measured in what we call millivolts, small m, big V. And what comes out of your wall, if you plug your phone into it to charge, is voltage, or V, big V. So a millivolt is 1/1000th of a volt. And out of your wall is typically about 110 volts. So we're orders of magnitude, or actually 3 orders of magnitude below what comes out of the wall. Okay, so we're talking about very, very small voltage, but they can be energized with electricity, with ions, positive charged ions or negative charged ions. So that's the neuron. Well, we have more other cells, and these other cells are known as neuroglial cells, and they're supporting cells. We refer to them in short as glial cells, and they're 9 to 10 times more prevalent than neurons. We have 4 different types in the central nervous system, and we're going to walk through them. And then we have 2 different types in the peripheral nervous system. So let's just take a peek. So same picture, different text. We're focusing on central nervous system. Astrocytes. Astrocytes are the most abundant. You can see a picture right here and where my cursor is pointing. And the astrocyte, as the most abundant glial cell in the central nervous system, plays a role in a couple of ways. It helps to support and brace the neuron. You can kind of, I think, appreciate how the appendages of the astrocyte are almost like reinforcing this neuron right here, this brown structure with a nucleus. And this astrocyte braces it or supports it, and actually also connects it to a nearby vessel or a capillary. So the second role of an astrocyte is for exchange with capillaries, where we're going to have gas exchange, oxygen, carbon dioxide, to support metabolism, removal of waste products. And during development in the central nervous system, the astrocytes help to guide the growing neuron, in utero. That's a third role. And then like a fourth role is really under the category of metabolic cleanup, where it helps to recycle neurotransmitters. And we'll talk more about neurotransmitters, but they're chemical signals that help link the transfer of a action potential or a signal of electricity from one cell to another, or from one neuron to another neuron. Because when two neurons get together, there's a little bit of a gap, a very, very small gap, and the neurotransmitter crosses that gap, okay? All right, next cell, microglial cell. Microglial cells are like small macrophages right here. Here's one, here's the second one. And they operate to kind of clean up debris that might be found within the central nervous system. So if a patient has a stroke and there's damaged central nervous system tissue in the brain, the microglial cells eventually will show up and try to clean up the damaged tissue, and they lay down repaired tissue, which repaired is from the wound healing lectures that we talked about, has a scar or has fibrosis because the function of that brain tissue no longer works. Just like a scar on the skin, when you get a scar in the brain, now you've lost functionality of that portion of the brain. Well, the microglial cells operate like macrophages in the skin. Ependymal cells are third type. They line the central cavities of the brain and the spinal cord in the ventricle system. And they're responsible for the production of cerebral spinal fluid. And so the central nervous system, the brain and the spinal cord are bathed in this cerebral spinal fluid, and it's filtered blood. It has a consistency of like blood plasma. It's clear, slightly yellow, but no red blood cells, so there's no red appearance. And there's really no cells in there. It's mostly a protein fluid. And it's filtered at the ventricular system in the central nervous system through these ependymal cells that form a permeable barrier for the production of cerebral spinal fluid. Okay, our last one, oligodendrocytes. You can see right here and right here and right here. These are the ones that actually manufacture what we call myelin sheaths. In myelin sheaths, we're going to get into it in a little bit more detail later in the lecture, and even in subsequent lectures in this unit. And really the thinking around myelin sheath, or the way to, I think, visualize it is basically a fat with protein inserted into it because it's synonymous with the plasma membrane or the cell membrane. And the oligodendrocyte makes extra membrane and then uses that to wrap the axon. And the reason it wraps it is to help facilitate faster electrical conduction. And we'll talk about that in subsequent lectures. So right now, just kind of pause and set that on the back burner on how the myelin sheath actually helps to facilitate faster action potential, because we're going to go over that in detail. So these are the 4 glial cells in the central nervous system. Now let's look at glial cells in the peripheral nervous system. It's even simpler. There's only 2. We've got satellite cells and we've got Schwann cells. Now, the satellite cells provide electrical connection, or help to regulate, that's probably a better way of saying it. They help to regulate the environment around the neuron. So in a way, these satellite cells operate very much like an astrocyte in the central nervous system. Okay, these satellite cells help to provide nutrition and support for the neuron in the peripheral nervous system. And then the second one are Schwann cells, and Schwann cells are responsible for manufacturing myelin sheath. So the Schwann cell in the periphery behaves a lot like the oligodendrocyte. Okay. So you can appreciate that in the periphery, we don't have microglia, we don't have epidymal. Well, in the periphery, we have macrophages, so we don't need microglia. And the epidymal cells are really to produce central nervous system fluid, cerebral spinal fluid, excuse me. So you don't need those in the periphery. So here's a Schwann cell. This is a peripheral neuron, and you can see this branch right here, an axon collateral or side branch. This arrow is a blow-up diagram of what's happening here. And this is the axon, this sort of tube down the middle. And the Schwann cell operates like an oligodendrocyte, where it's manufacturing extra cell membrane and literally just wraps around the axon, okay, as it grows. So it's a living cell that makes extra plasma membrane, but between one Schwann cell and the next, there's a little bit of a gap. So you have a myelin sheath, a gap, and then a myelin sheath. So let's look at this one. Myelin sheath, gap, myelin sheath, gap, myelin sheath. Okay, and we'll talk about the purpose of these intermittent gaps, or what we call nodes, internodes. Later, we'll talk about those later. So the myelin sheath, it's a protein lipid filled cytoplasm of Schwann cells, like a membrane. And the neurolemma is the outermost part that contains the nucleus of that particular cell, and the cytoplasm. And the myelin sheath is the inner layer of the plasma membrane. It helps to protect and insulate the axon, just axons, never dendrites, just only axons. And it helps to propagate a faster transmission of the action potential, or this electrical signal. And we'll show how that happens mechanistically. We're just going kind of an overview today. But the gaps in between that we talked about, in between the myelin sheath, are what we refer to as nodes of Ranvier. Ranvier. This was a French individual, so it's more of a French pronunciation of Ranvier versus Ranvier, right? That's how an American would say it, Ranvier. Ranvier. Oligodendrocytes serve the same purpose in the central nervous system, like we talked about. And this schematic tries to help explain how this works. So you've got the Schwann cell, and I know this is a silly example, but I remember as a kid, my mom used to make this hot dog dish. It was called Pigs in a Blanket, and she'd get like hot dogs, and then get like those tubes of crescent roll that you buy, and you peel it off, and then it pops, and just really good, make really good like croissants. And she would wrap the hot dog with this croissant dough, and then bake them, and call them pigs in a blanket. And the way that you would make it is you kind of roll the hot dog in this croissant pastry, if you will. So that's what I think of when I think of how the myelin sheath is made. So this Schwann cell makes extra membrane and literally just wraps it around and around, and it's at some level like this pigs in a blanket, or maybe like a cinnamon roll, but you're wrapping it around the actual axon. It's kind of cool. So another purpose of the myelin sheath in the periphery only. So first goal, or first purpose of the myelin sheath, is to help facilitate or speed up action potential. And that happens both in the central nervous system and the peripheral nervous system. But in the peripheral nervous system only, there's a second value or purpose for having it. Well, they form what we refer to as a regeneration tube. And this helps for healing of peripheral nerves. Now, careful, just peripheral nerves. Central nervous system nerves do not heal. They do not regenerate. So this is important, and you can probably appreciate this clinically. If you have a patient that has a spinal cord injury, let's say it's say about C7 or C6, which is above the plexus, that it's in the neck, above the plexus that actually perfuses or innervates the arms, then they're going to be paralyzed from the neck down forever. And they won't be able to regenerate those nerves or heal. And individuals that, you might know that this has happened to, unfortunately, maybe in your family or friends, but Christopher Reeves was like a famous actor, the original Superman, who fell off a horse and had a neck fracture, and his central nervous system nerves never regenerated. And then if it's further down on the spinal cord, you might have, instead of quadriplegia, you might have paralysis of the lower extremities, and you might have use of your hands if it's below the plexus that actually goes and innervates the arms. So this is actually a pretty important concept. The central nervous system nerves do not regenerate, but in the periphery, we do have the ability for regeneration of nerves. And the way that this happens specifically with myelinated neurons is you can see this first image is a normal nerve fiber. Here's local trauma. We've got macrophages, not microglial cells, because this is the periphery. And this is a muscle fiber that it's innervating. So it's a nerve innervating onto a muscle fiber. Well, that muscle fiber starts to shrink. And you can kind of appreciate the size of this muscle fiber here is thicker than here. And now it's even thinner because it's not innervated. And you get atrophy of a muscle when it's deinnervated. Well, what happens here is the myelin sheath is going to start to extend and grow. And so in this next picture, you can appreciate how the Schwann cells in the periphery, the Schwann cells form this regeneration tube, and this muscle is continuing to atrophy in this cartoon image. It's actually a very accurate image. It's one of the reasons I like it. But you've got a new regenerating axon that eventually will reinnervate that muscle fiber and now reestablish the ability for that fiber to, or that muscle to contract. So I'm setting the groundwork for innervation. Signal is sent from the nerve down its axon, innervates onto a muscle. We're going to call this interface right here a neuromuscular junction. We'll get to that later after exam 3 in unit 4. And this is what sets the stage for being able to fire a muscle. So first you have to have a signal that makes it to the muscle to allow it to contract. And this is actually a fundamental reason that I select in my courses to teach nerve physiology first and then muscle physiology second, because you can't have muscle contraction unless you have a signal from a nerve telling it to contract. That's just the way it works. Okay. All right. So what are some details, about neurons? Right? What about neurons? What are some details? Well, they tend to be long living as long as there's not trauma or damage. But they don't divide. They are amitotic is the name for that. Amitotic. The exception is actually olfactory and memory neurons that actually have the ability for division, cellular division. Olfaction and memory. Seems like kind of two weird exceptions, but we'll talk about this later when we talk about memory and the central nervous system in unit 4. But those two processes are actually very closely linked. So let me just digress for a second. I want you to think about a really valuable memory of yours, a positive memory, one that makes you smile. Okay, maybe it's, like your grandmother, or maybe your mom or your dad or a sibling, or a place, a location. And now I want you to kind of close your eyes and try to think about a smell associated with that memory. And let's say, for example, for me, one of my memories is my grandma. And there are certain foods that I smell. I can walk into a kitchen, smell a certain food, and instantly be reminded of my grandmother because she used to make those foods for me. And so memory and olfaction are very closely linked. And we'll talk about that more in the central nervous system, but those centers are actually helping to communicate with each other through interneurons that we mentioned in the beginning of the lecture. Okay, now back to general things about neurons. So the metabolic rate. The metabolic rate is actually very high for neurons. They're constantly working. And this is why, I don't know if you've noticed this, but if you're studying, hopefully you've noticed this, okay? If you're studying for my exams and you study for hours, you're probably hungry when you're done. And you're like, why am I so hungry? I didn't do anything today physically. I didn't work out. I just studied all day for Dr. Keller's crazy exams. What is going on? Well, neuronal activity, like when you're really into a project and you're using, your central nervous system, a lot of neurons there, you're exercising, and the concept of exercising your brain is real. Metabolic rate is actually quite high for the nervous system. And when you're in a thinking mode, you burn a lot of calories, okay? Now, these have bundles of arm-like processes, So look at this. We've got these dendrites up here that receive the signal, goes to the cell body, and then it's sent down these axons to other neurons or to muscles in the periphery. So what is that all about? And why do we do that? Well, we set up these pathways and we call them tracks in the central nervous system. And we call them nerves in the peripheral nervous system because they're going somewhere. So in the literature, like in your textbook, or when you're reading the literature, if I give you another peer-reviewed paper for a bonus assignment, you're reading that manuscript. If it talks about tracks, you're in the central nervous system. If it talks about nerves, you're typically in the periphery. So let's dissect the parts of a neuron. And, I told you that this is just a cell. It's a specialized cell and it looks different than maybe a normal cell would look, or quote unquote normal cell, which you kind of think is looking like a fried egg, right? Where it's kind of got like a ovalish like shape. And then there's the yolk is the cell body. Maybe the cell body is pushed over to one edge, or maybe it's right in the center. That's what you think of when you think of a cell. Well, this is a cell. I mean, this is the fried egg, right? This is the main part of the cell, and here's the nucleus. But it has all these specialized features. But it also contains a lot of components, just like what you see in a standard run-of-the-mill cell. All right, and I'm trying to get you prepared for muscle physiology, because when we go into muscles, we're going to talk about a myocyte, like a skeletal muscle or a cardiac muscle or even a smooth muscle. And they also are just specialized cells. So the cell parts that we're looking at, we've got the soma, or what we refer to as the cell body. It's got all the normal organelles you would expect: mitochondria, a nucleus. It's got endoplasmic reticulum. It's got a Golgi apparatus. The only thing that is missing are centrioles, because it doesn't divide, right? So it doesn't need division parts. The Nissl bodies. The Nissl bodies, it was named after an individual who discovered it, but it's basically rough endoplasmic reticulum. Helps for protein manufacture. The nuclei, when you get a cluster of cell bodies in the central nervous system, you're going to call them nuclei. When you get a cluster of these cell bodies in the peripheral nervous system, you call them ganglia. So this first blank is central nervous system, and the second blank is peripheral nervous system. It has dendrites, specialized features known as dendrites that receive a signal from another axon. And then the axon, this component right here that looks like a funnel is actually referred to as the axon hillock. It's part of what we refer to as the trigger zone. Trigger zone, like it suggests, preparing for the axon to fire. And when we say an axon to fire, we mean send an electrical signal. That's what we're saying. Now, you've got these axons. You may have an axon collateral. This is a little bit more rare. You don't usually have a side branch on every axon, but you can. And we talked at length about the myelin sheath and the gaps in between the myelin, the nodes of Ranvier, and how they, help to allow in the periphery for them to regenerate, forming a regeneration tube, or in the central nervous system and in the periphery allow for a more speedy signal being sent. Well, where's the signal being sent? Well, it's being sent to another nerve or a target organ. Now, we also have down here towards the bottom, we've got these terminal arborizations. And those are part of the terminal branches, and then they end on a synaptic knob. And the end of the nerve kind of comes here, and then it bulges at the very tip, kind of like, and they call it a button because it kind of looks like a button. I'll show you a picture of it here in a second. And then two other terms, an axoplasm. Axoplasm is the cytoplasm. This is the same thing as the cytoplasm in the cell. Okay. And then axolemma. The axolemma is the cell membrane that surrounds an axon. They just have specialized names because it's a specialized cell. Now, if we're at the very end here, and I told you how this terminates into smaller and smaller little branches, and then it bulges. And here's a picture of that synaptic knob right here. So it looks like a button, doesn't it? So you can see all these synaptic knobs, and what are they like inserting into? Well, they're not really inserting. They're just really, really close. There's a little bit of a gap. I don't know if you can see my fingers here. Here's the knob right here, and then here is the surface that it's innervating on, and there's a little bit of a gap. And we'll talk more about that gap, or that synapse is what we refer to it as later. But right now, I just want you to be able to appreciate that this is actually what we call a synaptic knob. Here's the axon leading to the synaptic knob, and the synapse is that gap. So if we're on this side of it, we call it a presynaptic neuron. And if you're on the other side of the gap, we call it a postsynapse. So this is the postsynaptic neuron right here. And it's the cell body. This kind of big bulgy thing is this cell body of the other side of that gap from one neuron to another. Make sense conceptually? Okay. What kind of neurons are there? Well, we can classify them by structure. We can classify them by function. So if we talk about structure classifications, it's how they're organized. So we either have multipolar neurons. We have bipolar. So multipolar, where you can see that you've got many different dendrites. I mean, look at this one. This one's like crazy. Whereas a bipolar, it's got the cell body here. You got one axon, and you got one branch that leaves. So bipolar too. Unipolar, where the cell body is kind of off to the side. So you just have kind of a single axon, and then there's sort of this cul-de-sac, if you will. So these are structural classifications of neurons by architecture or the way they're organized. Now, what about functional classifications? This is a previous picture that we looked at in the very beginning of the lecture, but it paints the picture that I'm trying to portray, which is you can classify neurons instead of by structure, either by function, sensory, afferent, motor, efferent, or association neurons, interneurons. Okay? So again, if I gave you, hypothetically, a bonus opportunity, like we did in Unit 2, where you're reading a paper from the literature, which I actually love these kinds of bonus opportunities, because if you're willing to do some extra work, I'm willing to reward you for it. I think it's going to help you in the future, because being able to read a textbook is one thing. Being able to listen to my lectures is one thing, but being able to read a manuscript from really bright people in their field, in the language that they would talk to colleagues. If you read peer-reviewed papers, you're reading experts writing. And if you can understand that and you can actually synthesize information from it, you're operating at a higher level. You really are. And that's pretty impressive. So if I gave you another manuscript and you see words like sensory or afferent, you know it's a functional classification, or motor, efferent, or interneurons, association neurons. But if I gave you a paper and they talked about these bipolar neurons or these unipolar neurons, you know you're classifying it by architecture or structure. Does that make sense, the difference? I know you can't really answer me, so I don't know why I'm pausing, but it's just habit. All right, we're getting into the portion of the lecture that I was going to warn you about, right? That I suggested I'd warn you about. So what I want to do here is I'm gonna pause my screen share and just kind of give you a little bit of a pep talk. Typically with action potentials, nerve physiology, students need to listen to this, to read this, to understand it a few times before it really sinks in. This is one of the more complicated concepts within this course, is nerve physiology and action potential. Another very complicated topic that we'll get into later in the semester is muscle contraction. And I'll have the same kind of, heart to heart with you. So we're going to dive into it now. But again, if it is a little bit over your head, don't be surprised. Don't be scared. The beauty of recorded lectures, whether it's live or, on an office recording like this, is you can rewatch it. But I want to encourage you to ask questions. Come see me in my office hours. If they don't work for you, let's schedule a time that does. Okay. And we might have to get overly creative. We could do it on Zoom if you're just not getting this information. Okay, fair? Let me reshare my screen and let's dive into neurophysiology. And we're going to go over this a few times. This isn't the only time we're going to do it, but this is the setup. So we are looking at the membrane. And if you remember back to Unit 1, and you're saying, I'm trying hard to put Unit 1 behind me. Well, this course is cumulative because life is cumulative. I'll be honest with you. I mean, if you learn something in life and then you have an exam, meaning you have a life event that you needed that information, and then you oh, phew, I'm so glad I learned how to deal with that situation. Guess what? You're gonna have that situation come at you again. And the next time it comes at you, you're going to be even more prepared. And if you want to go into research or clinical medicine, these are concepts that you can't forget. They just continue to build. So we're going to try to refresh a lot of these concepts that not only did we review it in Unit 1, so it's review from this course, but it was part of your prereq on, the cellular biology, okay, that comes out of basic bio courses. So that all being said, let's review. Hopefully this is familiar. If it's not, look back at Unit 1. And this is the phospholipid bilayer where my cursor is. So we've got the tails pointed in and we've got the heads pointed out. And we have transmembrane proteins, like in orange, this big sodium channel in orange. And then this one right here in blue, this big potassium channel. And so you can appreciate the potassium ions are leaving through this channel. And the sodium ions look like they're coming into the cell through this channel. Because these are actually leak channels. These are channels that kind of are always open, allowing potassium to go from a high concentration at 150 milliequivalent per liter down its concentration gradient to 4 milliequivalent per liter. So it's going down its concentration gradient through diffusion and leaving the cell. And sodium, this is the extracellular fluid. And in here is the intracellular fluid. This is inside the cell. These clumps are large, negatively charged proteins, anions, that are too big to leave. So these large negative proteins help to maintain a slight negative environment inside the cell. Not like negative, like Debbie Downer or something like that, but negative charge. Okay. So setting this up, we've got sodium inside the cell that's low, sodium outside the cell that's high. We also have a sodium potassium pump that's not shown on this picture, but that pumps 3 sodiums out and 2 potassiums in. And that also helps to maintain a slight negative environment inside the cell. We'll talk about that later. But this is sort of the baseline of a cell. And the baseline of a cell is that it's slightly negative inside, mostly because of these large negative proteins. DNA, RNA are both negatively charged. There's a lot of DNA and RNA inside the cell. So the cell environment is a slight negative charge. Now, think through this conceptually. If I was to open a channel and allow sodium to flood into the cell that's carrying a positive charge, what would happen to the inside of the cell? Let me repeat that. If the inside of the cell at rest is slightly negative, it's about minus 70 millivolts, and I open a channel and I allow, through diffusion, a lot of these sodium ions to come in, carrying a positive charge, what happens to the charge inside the cell from minus 70 millivolts? The answer is, you can pause the lecture if you want to think about it some more before I tell the answer, but the answer is it increases the charge inside the cell, makes it less negative. It goes slightly towards the positive. Now, it may not become overly positive. It may go slightly past 0, but at minus 70, so if 0 is here and minus 70 is here, it might go a little bit past 0 and then it'll come back down, and we'll talk more about that. I just wanted to lay some of the framework for how we're going to use this information. So let's just review. Let's look at negative proteins, negatively charged proteins inside the cell, along with RNA, DNA. We've got a high potassium concentration inside the cell. We have a low sodium concentration inside the cell, and we have a low chloride ion, which is negative inside the cell. This is all in relationship to outside the cell. Outside, it's the flip. Low positive or low potassium, high sodium, and high chloride. Again, in comparison to inside the cell. Okay? All right, let's look at what goes across. So what goes across? Again, same setup here. We've got extracellular fluid up top, intracellular fluid down low. And this schematic is only showing or only broadcasting the sodium, it's highlighting sodium and what sodium's doing. And so this is what I was referring to when I said, hey, what would happen if I opened a channel and I allowed a flood of sodium to come in? Because remember back here, sodium is high outside the cell, sodium is low inside the cell. So if I open a sodium channel, which way is it going to go? By diffusion into the cell, bringing sodium that's positively charged. Okay, so we have a couple of different channels that we want to cover here that are in the membrane. Now, where are we? Where is this cartoon? Well, if you think about our axon here, I'm sorry, our neuron, here's our axon, here's our cell body. We're right here where this box is, and we're blowing that up at the plasma membrane or the axonema. We're at the membrane. And the direction of the voltage or current is what that means, not like water current, but voltage current is from left to right. You go from the dendrite towards the axon. And we've got either two kinds of channels. We have a passive leak channel, or we have an active gated channel. Now, a leak channel is like a sodium channel, like here on the previous slide. This is potassium. This is a potassium leak channel. That's a classic example. It's always open. Okay. It allows ions to flow. This potassium, K plus, is the symbol for potassium. Potassium leak channel allows ions to flow from an area of high concentration inside the cell to an area of low concentration outside the cell. It's always open. It's always leaking. Now, what brings it back in? Well, remember, there's a sodium-potassium ATPase pump that we talked about in Unit 1. It pumps 3 sodiums out for every 2 potassiums that come into the cell. So the sodium-potassium ATPase pump brings this potassium ion back into the cell. But the leak channel is always open. Now, a gated channel or active channel is either ligand gated, something binds to it and then opens it, like what you're seeing right here with this sodium active gated channel. You've got a ligand binding to it and opens the gate. Or it could be voltage gated, which is setting us up for sending an electrical signal or a current, as we show here, from the left to the right. So the resting membrane potential, what is it? Well, it's -70 inside neurons. It's the resting electrical charge or membrane potential. The word potential means there's a difference, an electrical difference between inside the cell to outside the cell. So you measure the resting membrane potential by putting an electrode in the cell, and then you put an electrode outside the cell and you measure the delta. But it's a very, very small difference, and it really only exists at the plasma membrane, at the surface. If you move deep into the cell, away from the membrane surface, it's pretty much a neutral charge. It's really a micro, small, local electrical difference. Okay? So what causes it? Well, we've already kind of talked about some of these things, right? We've talked about this same picture that we looked at before. We've got the permeability, or this leak channel. It's potassium permeable because this potassium channel is always open, allowing potassium to leave, taking positive charges out. And it's not sodium permeable, so it doesn't, necessarily allow for sodium come in. You actually have to have a ligand or a voltage, or you have to have the sodium-potassium ATPase pump running. And the sodium-potassium ATPase pump, to review, it takes 3 sodiums out of the cell, and it brings in 2 potassiums for every ATP that it uses. Now, in addition to that, we've got a tremendous amount of protein, RNA, and DNA inside the cell that really establishes the resting membrane potential to be slightly negative. The sodium potassium pump helps to maintain that slight negativity at the plasma interface. But I also just want to point out, this is a rather inefficient process. Like, you're always leaking potassium, right? And then you got to run the sodium potassium pump to reestablish the ion balance. So the nervous system uses 20% of all the energy in the body, even while you're sleeping. So imagine when you're studying for an A&P exam for Keller, how much energy you're burning, right? That's why you need valuable, good quality snacks. Okay. Let me show you this video real quick, because I think it will help with the visualization of what's going on with the sodium potassium ATPase pump. So there's no audio on this. I just want you to be able to watch what's going on. This is our plasma membrane, sodium potassium ATPase. This is inside the cell down here, outside the cell up top. 3 sodiums bind. They dump out into the outside environment. There's your 3 sodiums out of the cell. Now look, you're going to see potassium. Now that this transporter is open to the outside, you get 2 potassiums that bind. You get a conformational change of the pump, and it brings 2 potassiums into the cell. Wait for it. There it is. Make sense? Very simplistic. Here it is again. 3 sodiums are binding. Here's your ATP binding down here. You cleave the phosphate. That's the energy required to run this thing. It's literally a pump, moving ions one direction and other ions the other direction. And if it's not super clear on the video, you can go to the link that I had in my slides. Okay? Hopefully that helps. All right, let me get back to where we were. Right here. Okay. So sodium potassium ATPase pump. And that's the link. What causes changes in membrane potential? Right? In the remaining slides here, we're almost finished. We have about 5 or 6 slides. I'm trying to set up our next lecture, which is talking about action potential and these membrane changes. And so the action potential changes because membrane permeability to certain ion changes. You allow for sodium to come in. When at rest, it's impermeable to sodium. The only way sodium comes into the cell is through the sodium potassium ATPase, or a ligand that binds and brings it in. So now sodium floods into the cell if you open a sodium channel, and you can either form what's called a graded potential or an action potential. A graded potential is a potential, again, we're using the term potential as a difference in electrical charge that's over short distances or small. An action potential is one that travels the entire length. Okay, that's the difference. And from this picture that's shown here, you can kind of appreciate, here's an unmyelinated axon, and then 2 myelinated axons, right? And the reason this is here is I'm really trying to highlight the speed of this electrical signal, if it's a graded or an action potential. Because if it's an unmyelinated, look at the speed in meters per second. 0.5 to 2 meters per second. So in 1 second, you can go 2 meters, right? That's about 6 feet in a second. Okay, that's how fast. Now, look at this. You can go, if you myelinate it, you can go in, you can go 12 to 30 meters in a second. So 30 meters, so 30 meters is almost, it's not quite, it's about a third of the distance of a football field, right? Right, so that's pretty far. Like you can go from the goal line to like the 30, it's actually probably more like the 35-yard line in 1 second versus 6 feet in 1 second. It's a big difference. Okay, so we're saying that what causes a ability for the membrane potential to change? A change in the permeability of the ions. So we're going to be looking at pictures like this in our next lecture quite a bit. And we've got membrane potential. We get a reduction in membrane potential where there's less difference between the inside and the outside of a cell under what we call a depolarization event. The membrane becomes depolarized. We're saying less polarized. So the word polarization, like if you're polarized on an issue with a friend, you think one way, they think another way. There's a big difference. You're polarized. Depolarized means that you think, you close the gap. So when we say membrane becomes depolarized or a neuron becomes depolarized, we reduce the membrane potential. We have less of a difference between the inside and the outside of the cell. So the cells become less negative. So instead of minus 70 millivolts, when we depolarize, we go, here's 0 right here. These are millivolts. This is the y-axis. Over time on the x-axis, you go from minus 70 towards 0, less polarized. Makes sense? And then we're going to hit what we call threshold. We'll talk about that more later, next lecture. And you fire this action potential. Then you repolarize. And you might overshoot as channels are starting to close to reestablish your resting membrane potential. So right now what we're talking about is resting membrane potential and a depolarization event where you become a local potential or a graded potential that brings you towards threshold. Once you cross threshold, you call it a depolarization event. You fire the neuron. And then here at 4, you repolarize, you move ions back to where they're supposed to be, reestablish the negativity, but these channels close not instantaneously. They close a little slow, like a garage door. And so you might overshoot a little bit and bring, in and out too many of the right ions. And that's what we call hyperpolarization, is where it becomes a little bit more negative than resting, and then it kind of settles back in. So hyperpolarization is opposite to depolarization. Depolarization's becoming less negative. Hyperpolarization is becoming more negative. But the other thing in a hyperpolarization event is you can't fire the neuron anymore. And so it's an inhibitory event to prevent the neuron from firing again right after it just fired, and until you reestablish resting membrane potential. Okay? And we'll go through this a few more times in the next lecture. So these local potentials, local potentials are also referred to as graded potentials. They can either depolarize or hyperpolarize, but they're very short-lived. They don't last long. They can be ligand regulated, something can bind, and the magnitude varies a lot with the strength of the stimulus. So for example, you can see here this graded potential, this one's going in a depolarization direction. This one's going in a hyperpolarization direction, showing you the options. And you can actually grow these or summate them on top of each other. And then if you look at the corresponding magnitude of the stimulus, So for example, if this is the potential result up top, and this is the timing of the stimulus, this one firing right after it, next to each other, you summate and you get a bigger response. Okay, these are local graded potentials. They can add up, they can summate, or they can be additive. So some initiators of local potentials: energy, heat, light, a neurotransmitter. Characteristics, well, they don't stick around for a long time. They fire and then they dissipate quickly. They are reversible. And they can either be hyperpolarization, which is more negative, or they can be depolarization, which is making the cell more positive. Okay? In our next lecture, we're going to talk quite a bit about action potentials and polarization events. And so this is really the foundational setup, along with your chapter 7 reading, to get you ready for next week. A couple of review questions before we finish up. Which of the following is not part of the central nervous system? And I'll pause here for a minute before I give you the answer, or you can pause too, the recording. The answer here is Schwann cell. Schwann cell is found in the peripheral nervous system. All right, last one. Sodium potassium pumps in neurons consume an estimated 20% of all energy in your body. Why? I'll pause here before I give you the answer. And you can pause the recording too. The answer is B. They're required to maintain resting membrane potential. So remember that the pumps, the ATPase pumps, don't establish resting membrane potential, but they do help maintain it. Kind of what establishes it is a lot of the negatively charged anions that are big, like proteins, RNA, DNA inside the cell. But the ATP pump is really important for maintaining that resting membrane potential. Okay, so today we talked through introduction to nerve physiology, kind of setting the groundwork for this unit. Looked at the functions of nerves. We compared and contrasted the central and peripheral nervous system. We looked at nerve anatomy, like the parts of a nerve. We talked about regeneration of nerves that really only happens in the periphery. And then we compared and contrasted resting and graded potentials. All right. Thanks so much for listening, and I'll see you soon.